Hi again, everyone. My name is Alois Mwambwiga. I'm here to introduce you the next speaker. By her name, Pamela Akuku. She's a PhD student at the IPES Institute in Spain. She'll be presenting here Animal Bones, the Keepers of History. You're most welcome, Pam. It's your time.
Thank you, Alois and Fergus, and thank you everyone for attending. So, like Alois said, my name is Pamela, and I'm going to be talking to you about animal bones in the archaeological context and what information they can provide to us in terms related to hominin and hominin studies. So, basic definition, what is zooarchaeology? It is the study of animal bones remains from archaeological sites. And by studying them, we can get information on various things regarding hominins, such as their subsistence behaviors, their hunting strategies, and other aspects such as environmental change. And by environmental change, I mean the change from the paleo environment to the present. Factors such as domestication of animals, as well as ceremonial use of animals, depending on whichever time period you're looking at. So this study enables us to infer what the animal to human relations in a given site or area was, as well as the use of the sites and the basic environments. So in zooarchaeology, I basically look at bones, but you can look at various things, such as shells, bones, and teeth. I believe Raquel talked about teeth that she has been looking at, skin, hair, hooves, as well as exoskeletons. And these things can give us various information about the given site. So what can we learn from animal bones? We can learn a lot of things, and we basically have a procedure or schedule. So first we identify and quantify them, and then from then we can learn things such as their age, their sex, the environments that they inhabited, the morphology, and then we start looking at taphonomic modifications, which I will talk about a little bit later. So identification, the first step would be to identify what bone you're looking at, and then the species. And this we do by the use of manuals, which show us what the bone is, and as well as can give illustrations, as you see on the top left, of different species and different morphologies. If you're lucky enough, you can be in a university or in a lab that has a modern comparative collection, and this gives you a better idea because you see the bones up close and you can familiarize yourself with the different skeletal elements as well as species. So after identification of what bone, you try to quantify it further by classifying it into sizes or size classes. This is only one of the methods you can. There's different ways from people providing from class 1 to 5 based on the weight. So this is just one way in which you can quantify them, from your microfauna, which is less than 4.5, to elephants, which are greater than 1,000 kilograms. You can quantify your bones further, and we use various methods again, but I only highlight 3 today, which is the NISP. This refers to your number of identified specimens. So you will only count the bones you can identify to elements. Some people use the weight. So you take the weight of your bones and then you compare to the volume of sediment that you excavated, and in this way you can kind of you can calculate the density, mass over volume, so the faunal density of a given site. So after using the NISP, we usually get the minimum number of individuals, which just refers to how many individuals are represented by the specimens, the number of specimens that you have, the number of skeletal elements. So NISP, as I said, is the number of identified specimens, and you count each bone or fragment as one unit. A good example is the skeleton of a brown rat, and you can see the various individual skeletal elements. So each one would be counted as one, and the total number would be your NISP. Now, like any other method of quantification, it is affected by some factors, and one of the factors that affects it is fragmentation. On your left, you do see a picture of some fragmented bones, and if you were to count this, you'd get the NISP of 4. In reality, these are 2 bones. Number 3 is the tibia. Number 1, 2, and 4 is a femur, and it's the femoral element of the same individual. So if it wasn't fragmented, your NISP would be 2 and not 4. When fragmentation is very high, as you can see on your right, then you wouldn't get an NISP, because NISP is only the identifiable specimens. MNI, which is quantifying the NISP into the number of individuals that are represented in your assemblage. So you look at the bones, the size, as well as the sides, left or right. And then you use this to get your MNI. So for instance, from our previous example, the skeletal elements that you would have found, the brown rat, would only be an MNI of one. So just like NISP, MNI is also affected by issues sometimes, and it could underestimate the actual number of individuals. That is, you could count more bones belonging to a same individual. It is affected by fragmentation, and fragmentation is what might cause the underestimation of actual individuals, because you try to account for the fragmentation. You could also get different results based on how you split your assemblage up there. For instance, the 3 bones that I showed you, the MNI could be 3 or 2. 3 in the sense that these are 2 left femurs, so ideally they represent 2 individuals, and then you have a mandible there. So if you decide that each of these 3 elements belongs to 3 different individuals, then your MNI would be 3. However, some people would allocate like the mandible to the femur if they're the same species and same size, and as such, your MNI would be 2. Apart from that, we can also look at the body parts that are represented in any given site, and this might give us information regarding the hominins or the site itself. So for instance, based on the elements that you find, you can speak of issues such as carcass utilization. That is how the given animal or resource is being used. Are they using the parts of the animal that will provide more caloric intake? You can also get information regarding butchery patterns. For example, if you do find cut marks on the distal elements, such as close to the phalanges or on the ribs, this may be alluding to disarticulation. If you find it on the long bones, it's usually associated with filleting. So it can also give you a bigger picture of how the hominins are not only using the animal, but how they're processing it. The bones that we find also tell us different things, such as age. We can get the sex of the animal. So certain animals have different morphologies based on gender, which we always call as sexual dimorphism. So for some antelopes, ram, sheep, you have species where the male gender have horns or antlers and the females don't. So when you find such a thing, you can tell, oh, I'm dealing with a male or female. Another very helpful thing is the pelvis. So when you do find a complete pelvis, you can usually use it to get the sex of the species that you're studying, because female pelvis are different from male, they're altered due to the birthing process, to accommodate for the birthing process. Then we can also get the age of the animal we are looking at. So one thing we look at is the tooth, and this diagram just shows you the development stroke eruption of the tooth, all the way from 10 months to 9 years of a goat. And you can see in the various stages, at one stage, it doesn't have a molar, it gets molars, the teeth get worn on the occlusal surface and they start falling out. So when you get this at an archaeological assemblage or site, it can enable you to know the age at death of the specimen you're looking at. Excuse me. Another thing that aids in age determination is long bones. So to your left is a fused long bone, metatarsal, and to your right is an infused one. This enables you to tell the age, because the infused one is usually a juvenile and the fused is adult. So it's another way of knowing the age of the specimens that you're looking at. So the next thing that the animal bones can tell us is morphology in terms of movement, as well as the diet. So different animal species obviously adapt to various forms of locomotion. They can be swimmers, they run, undulating in snakes, and this morphology can tell you about the environment. So we look at the bone, they're adapted in various ways. A good example for getting the environment is bovids. So bovids are a good proxy for environments, and we use them because they would be the most dominant at any given archaeological site, especially in African archaeological sites. So their morphology reflects the environment because they're very habitat specific. You can find your grazers, and this would allude to open environments, your browsers, which is kind of a mixed woodland bushland going on, and your browsers would be the bushland, and your mixed feeder would be mosaics of open grassland as well as bushland woodland presence. Movements of the animal is also inferred from the morphology itself. So this is a cheetah skeleton, and cheetahs are built for speed, so they're streamlined in various ways. So when you find this in any given site, you can kind of infer the environments because they're adapted to running, and you can run better in open areas than in closed. The same is if you found a giant, say, buffalo like a Pelorovis with very big horns, then you'd also allude open lands because it would be very uncomfortable to try and maneuver with the horns in a closed environment. The dental morphology alludes to the diet of any given species that you have. And in this way, we can classify our animals as either herbivores or as omnivores. So if they're herbivores, then we can kind of tell the kind of environment and food the kind of food they're consuming, and in this way, the type of environments that we expect and how it has changed. We can document that. And then we come to taphonomy, which is what I do. In the basic sense, taphonomy refers to the laws of burial, that is the study of the processes affecting the animal remains from death until recovery when we excavate a site. So we look at the bone modifications after all the identification and quantification. We analyze the surface for any modifications present. The modifications can usually be either natural, anthropogenic, or zoogenic. Natural modifications, so this is just a diagram showing you or trying to explain the process of taphonomy from death to, if it's stored, to movement such as fluvial transport, or if there is soil erosion, and then secondary storage, which may be it's being reburied, and then when we recover it. So the different type of modifications are the natural modifications, anthropogenic, and zoogenic. Natural modifications refer to modifications imposed on the bone from the natural elements of nature. So an anthropogenic refer to human stroke hominin modifications of the bone, and zoogenic is modifications that are found or impressed on the bone by other animals. So some examples of natural modifications include weathering. This refers to how long a bone has been exposed to the elements. It's just out in the open. And Berensma in 1978 conducted a couple of experiments, and in this way we have been able to document 6 different stages from 0 to 5, and 0 being less than a year of exposure for the bone, and stage 5 being a bone exposed to between 10 and over 25 years plus. So when you classify your bones into these different stages provided, you can get an idea or a picture of how long it was exposed before either being reburied again or even being recovered by us. These are examples of other natural taphonomic modifications. So the first picture is root etching. Sometimes when bones are buried, roots grow over them and they leave an impression. So that is etching, and that's a very good example of root etching that we sometimes find, which also gives us a better idea of what's happening at the site. Rounding, which is the alteration of the bone shape, stroke morphology, by the elements of nature. It could be by water, by the soil itself, and it results to the bone losing its original form in the most extreme cases of rounding. You also do have trampling. This would allude to the intensity of the site. Trampling can be caused by other creatures just basically trampling on the site, among other things, as well as it has been shown that Sand can also lead to abrasions on the bone, mimicking trampling. Polish or sheen caused by the rubbing of sediments on bone. It can be a natural occurrence, especially in sandy areas, in sandy sites. The next category is zoogenic modifications. So these are usually modifications that are placed on the bone by other animals. So I gave a good example of carnivore-based modifications. On the first slide is the tooth marks caused sometimes by carnivores. We call them scores, the linear tooth marks. You can also have digestion, which leads to alteration in the morphology of the bone. And this is one of the higher degrees of etching, acid etching or digestion in animal bones caused by hyenas. So this was from a hyena den. In the last picture, you see a very distinct circular mark, which is a puncture caused by a carnivore canine. So sometimes when we find this in the archaeological bone, we can see that they're also there are also other actors present in archaeological sites apart from humans. Before I go on to anthropogenic modification, sometimes I'm asked questions like, how can you tell that this was caused on a bone by an animal and human? How do you tell the differences? So we do have guiding principles as well, maybe from manuals as well as from past experiments. And the one thing they do say is that the imprint that tooth marks and cut marks leave on bones tend to be different shaped in the very basic sense of definition. Tooth marks tend to be more U-shaped, whereas cut marks tend to be more V-shaped. And from experimental studies, you can see the picture on your left. These are cut marks from experimental studies. And it has aided our analysis on bones because the picture on the right is from a bone in Buri. So initially it was classified as a cut mark and was alluded to anthropogenic modifications. However, Saleh et al. Studied it further and inferring from experimental studies, they actually found out that it is actually a crocodile tooth mark that left that imprint. So in a way, taphonomy is still a science that's developing, and through these experimental studies in conjunction with manuals, we can get more accurate results. We also look at fracture patterns. So the first thing we look at in a bone is if it's broken, is it a fresh or a dry break? So a fresh break happens when the bone still has, before the loss of significant organic fraction in terms of marrow, grease, whereas dry breaks would happen after a loss of the organic function or fraction. So after classifying them as either fresh or dry, we look at the specific fracture types, that is either a spiral, transverse, or longitudinal. And this informs us more on the actors of the bone and the actors of the fracture. So these are some pictures that show you clear examples. So the first would be a transverse fracture. And the one labeled C is spiral fractures. So we usually allude spiral fractures with hominins because this is what would happen when you're breaking a long bone to kind of access marrow and that bone grease. Anthropogenic modifications on bone, depending on the age of the site that you're looking at, you will get different anthropogenic modifications ranging from cut marks to burning in sites that are associated with hominin controlled fire. You also get owls. So this picture is from modern, but you have found there have been some archaeological owls found, for example, the Lascaux cave in France, and owls are used as needles. So you can see anthropogenic modifications directly. And the last one would be a Neanderthal bone flute. So these things can sort of give you information about the hominins. They were able to control fire. They were adapting and have complex behaviors such as cloth making, as well as music because of the bone flute. Other modifications would be bone tools. So this is in order of age. The first would be like pseudo bone tools from Pleistocene sites. And the next picture is from tools made from monkey canines from a late Pleistocene site around 45,000 years. And it's one of my favorite modifications ever because it shows real ingenuity. And then you also find bone harpoons, which is on the bottom left. And this is usually from Holocene sites that we do see bone harpoons. So this picture just shows you not only anthropogenic modifications, but when we find this in archaeological sites, you can kind of see the evolution in complexity. So in summary, why do we study all this is to understand the relations between the faunacetes and the humans. And in this way, I mean what prey-predator relations exist. Is there any competition with the carnivores? If in a site we are finding as many cut marks as we are finding tooth marks on different bones, or are we finding it on the same bone? Then if we are, then what is the order? Is it the carnivore access first? Is it the human access first? So by studying bones, we can kind of answer these questions. We can infer behavioral complexity, not only in terms of hunting and subsistence strategies, but as you've seen, there's complex behavior such as the making of owls for sewing clothes, as well as social aspects such as socialization, such as music. And we can recreate the paleo environment from the various taxa because, as we know from modern studies, that different taxas prefer or inhabit different environments. So when we find this, as well as conduct other things such as isotope studies, we can perfectly, well, not perfectly, but better recreate the environment. Like I say, taphonomy is a growing science. So there are two methods that I want to talk about that is current and developing that will better studies. The first is the use of zooms, which is not really new, but is relatively new. And in this way, we can better classify the taxa, the different species. So as long as you have your assemblage that preserve collagen, then you can conduct zoom studies, and then you can better get a taxonomic representation of your sites. The other method is called machine learning techniques. So what this does is you conduct your experimental studies and you feed the machine with information regarding the dimensions of things such as cut marks versus tooth marks, and it records this information. And then when you find this, or you suspect that you found this in your archaeological site, the machine can compare and give you by a 90% accuracy if you're looking at tooth marks versus cut marks, and give you a better representation of what's happening at your site. Thank you very much.
That was an excellent talk, Pam. Now we have a few questions here for you. I'll read them out first and then I'll post them in the Zoom chat so you can see them. So the first question,
okay.
The first question is, can you describe the reference collections you use, size of the collection, geography, etc.?
Okay, so currently, as I am based at IPES, we have a decent sized collection. It's complex in a bit in terms of there are not a lot of bovids represented here in Europe because it's a very European-centered collection. So with bovids, that's where it's a little crazy because we have a lot of bovid representation in Africa. But I do have a decent size comparative collection here.
Nice. Okay, so the next question, what is the leading department in taphonomic studies within East Africa?
The leading department with taphonomic studies in East Africa. I believe Mark alluded a little bit to this. We are a little bit lacking in this because even at the museum, which I'm kind of affiliated with, a lot of the studies focus on lithic studies. There isn't a lot of taphonomy. We do have a Zoom database and everything at the museum. However, with that being said, taphonomic studies
It could be
lagging
improved.
behind. Yeah,
I understand.
Yeah.
Okay, next question. Let me just post this in the chat. So For decades, taphonomists have argued quite aggressively, if I may say, over the meaning of early hominin assemblages. We still don't know if archaic hominins from East Africa scavenged or hunted animals. Why are we unable to solve this? This is a heavy-hitting one.
This is a heavy-hitting one.
Yeah.
Thank you. So I am aware that there has been a very heavy argument because it's the whole scavenging versus hunting. I think the only way we can solve this is through studying more assemblages and conducting more and more tests, like I said about the machine learning, and we can get better, accurate answers. So just increase our sample size globally. I mean, like archaeologists working in Africa versus Europe versus Asia versus Australia, and looking at same age sites. I think that's the only way we can solve this, personally, for me.
Okay, great. Next question.
Yeah.
Why do researchers ignore microfauna? What is the environmental potential of microfauna?
Oh, so I do not ignore microfauna. I'm actually looking at both microfauna and macrofauna. It's just today's talk was just a basic overview introduction of how we use animal bones. Microfauna has the greatest potential in terms of paleo-environmental recreation. So if you want to do that, then microfauna would be the best way. So I think it's up and coming, but more and more researchers need to look at it.
Okay, this is the last one that we have currently. Is there a future for zoo archaeology by mass spectrometry in East Africa? Many of the collections in local museums may be identified even if the size or morphology of the bone are not complete.
Wow. Okay, so I guess I would say if you're looking at a later ages of sites in Africa where you do find a lot of fragmentation and you're unable to resolve not only species but actual elements, like what is this? And if the elements preserve collagen, so if your site has good preservation, then yes, there would be a future for zooms. But if not, then you have the answer in your question.
All right, great. I'm looking here. I don't see any more questions. So I'd just like to thank you again, Pam, for giving an excellent talk and staying up very late, may I add, to give this talk. So I really appreciate it. And it was great seeing you again. And
It's good to see you too. Thank you.
And we will see the rest of you in the next session. Thank you very much for joining us.